This is part 1 of the lecture notes on viruses for Bio 112. The most important thing to learn about viruses at first is that they are not living things. They have some characteristics in common with living things, and we study viruses not because they are living things, but because they have some characteristics of living organisms, and they affect living organisms. They are parasites to them. They're pathogens, which it means they cause disease. So the viruses are not made, they're not cells. They're not unicellular, they're not multicellular. They are what we call acellular. Sorry, give me just a second. They are what we call acellular, which means non-cellular. That means viruses are not cellular. Viruses are parasites, and they can be parasites to animal cells, plant cells, bacterial cells. A great diversity of living things can be infected with viruses. What you're looking at on the left bottom of the page is a transmission electron microscope image. So transmission electron micrograph is what the image is called, of several different individual tobacco mosaic virions. A virion is an individual virus particle. So this one, for example, right here, is a tobacco mosaic virion that causes tobacco mosaic disease in plants. And it's a good time to talk about the name of the virus versus the name of the disease. In this case, the tobacco mosaic virus, or TMV, is the name of the virus, and the disease it causes is called tobacco mosaic disease, or tobacco mosaic virus disease. But sometimes the virus is not named for the disease it causes, especially in situations like the common cold or the flu, especially the common cold, because the common cold can be caused by rhinoviruses or adenoviruses. So many different types of viruses can cause common cold symptoms that we don't name the virus after the disease. But what you're looking at on the right is interesting because these are orchid leaves that have been infected with tobacco mosaic virus. Tobacco mosaic virus can infect plants, tobacco of course, and tomatoes, and orchids, and many other different types. This is showing you leaves in various stages of decay. I'm not sure that they're in order because the leaf would not go from yellow back to green. So I'm assuming that this would be the first stage of decay. You start seeing some brown spots, and then number 2, more of the leaf is turning a darker color. Number 3, the leaf is now composed of only yellow pigment. The green chlorophyll pigment is gone, and there are brown spots covering the leaf. And then 4, the leaf has turned entirely brown. So these are just different stages of the tobacco mosaic virus in orchid leaves. Are viruses alive? We've answered that question. No, viruses are not alive. Viruses do have nucleic acid, either DNA or RNA, and they do have an outside capsule or capsid made of proteins, and some of them are actually surrounded by phospholipid bilayer envelope. But and even though they are able to reproduce, they can't reproduce on their own. They are When viruses replicate or reproduce, they are entirely dependent on their host cells. If they didn't have a host cell, they could not reproduce. So they also do not metabolize and they do not grow. When they are assembled by their host cell, they don't change after that. The virus itself doesn't change after that. Now, when viruses reproduce, the new viruses can be mutated forms of the old virus. But once a virus is actually assembled, it's not going to grow or change in any way. Metabolized, just remember from Bio 111 that metabolism is all the chemical reactions that occur in our cells. So viruses do not metabolize. They don't carry out aerobic respiration or anaerobic respiration. They don't convert oxygen and glucose into carbon dioxide, water, and ATP. You know, they do not carry out those types of reactions. So, and also because they are non-cellular, we can answer this question, are viruses alive? We can say no, because there's just too many things that living things have that viruses do not. Viruses weren't officially discovered until the 1930s, when scientists could actually see them under an electron microscope. These are transmission electron micrographs, and they show they show you visually some very important things. Look to the picture on the right, picture B first. What you're looking at is human cells that line the intestine. So here is one cell that's lining the intestine. And here's the nucleus, which you can make out pretty easily. So this is a human cell. And inside the intestines, in the space inside the intestines, are some particles that look like this. And this is an E. coli bacterium. So that's the size of a bacterium compared to a human cell. Then, if you look back at picture A, this is just a partial image of an E. coli bacterium, and this is the size of a virus that infects that E. coli. So viruses are very, very small. Just imagine this virus on the surface of the E. coli. So viruses are so small that we do have to have special microscopes to see them. Transmission electron microscopes and scanning electron microscopes. But they were actually known about before they were seen. And the reason for that is that two different scientists showed that the virus could be transmitted. They couldn't see the virus, but the disease could be transmitted even when samples had been passed through a filter that filters out bacteria. The disease was still spread, and that let them know that there's something else out there that causes disease, causes sickness. So we're going to talk a little bit about the differences of the viruses, but first I'm going to cross this word out. These are not theories, these are hypotheses. But there are three hypotheses on the origin of viruses. We really don't know for sure, but the devolution hypothesis, also called the regressive hypothesis, says that viruses evolved from free living cells. That means a cell like either a bacterial cell or a eukaryotic cell of some kind. The virus evolved from that cell, and that's devolution or de-evolution, because the living cell evolved first and then the virus evolved, kind of like backwards evolution, because a virus is so much simpler than a living cell. Okay, then there's the escapist hypothesis or progressive hypothesis. And that just says that the RNA or the DNA core of the virus escaped from a host cell. And the self-replication hypothesis kind of reminds me of coevolution. Bees have evolved and pollinators have evolved along with flowers. The flowers have evolved colors and scents that attract pollinators, and the pollinators have evolved, body parts and skills that help them to find the flowers that are going to give them the nectar that they need. That's coevolution. The flower and the pollinator evolve together. So this is saying that the host cell and the virus evolve alongside each other. And there are studies of plant pathogens that support this hypothesis. But again, they're just hypotheses. Now we're going to talk about viral morphology, and morphology just means form or structure, the study of form or structure. An individual virus particle is called a virion. So if we go back to that first picture where I circled right here, this is a tobacco mosaic virion. That's an individual virion, and they are viruses when you look at all of them that are in that picture. So one virion always has a nucleic acid core, and that nucleic acid can be DNA or it can be RNA, and it can be single-stranded or double-stranded. And the chromosome itself can be linear, like human chromosomes, or circular, like bacterial chromosomes. But here's what's different about the nucleic acid core. We have learned up to this point that if we made a chart and compared RNA to DNA, one of the differences is that RNA is single-stranded, DNA is double-stranded. However, with viruses, the RNA can also be double-stranded and the DNA can be single-stranded. And this is going to be really important in how we classify viruses today. Okay, so the virus is going to contain a nucleic acid core, one of 4 different types: single-stranded RNA, single-stranded DNA, double-stranded RNA, or double-stranded DNA. And then outside that core is a capsid made of individual capsomeres. And sometimes outside the protein capsid is an envelope, which is very similar to the phospholipid bilayer in eukaryotic and prokaryotic cells. Contains a phospholipid bilayer with proteins embedded. So, viruses may also contain enzymes, their own enzymes and other proteins. And the genome of the virus, whether it be single-stranded RNA or DNA or double-stranded RNA or DNA, contain the genes to produce proteins that the virus can't get from the host cell. So a lot of times the virus will use genes from the host cell to do certain things, but sometimes the virus will have its own genes. Okay, so this is a lot to read. The main difference is that DNA viruses do not tend to mutate